This video is going to offer a quick overview of mitosis and meiosis. I have another video which goes through these topics in greater detail, including the stages of prophase in meiosis. But I think I teach different classes and each is appropriate for individual levels. And so this is a shorter, more generalized version. So if you were to look at a fish embryo, obviously the cells are dividing quickly as you go from this microscopic embryo to a young fish. And so as you look at this embryo, you can see there are stages where you can see a nucleus, and I don't really see chromosomes. But then here, I really don't see a nucleus anymore. At least there's no nuclear membrane, but I'm starting to see chromosomes. They don't look very organized. I see these two areas on either side of this region of the chromosomes. And it looks as if these chromosomes are beginning to become organized. There are stages where the chromosomes seem to align in the middle of a cell. Then there seem to be two sets of chromosomes moving in opposite directions. And then ultimately, then we go back to where we started. So there seems to be a process here, a predictable process which one sees over and over again. Now, later in this video, I'll get back to the idea of chromosomes and counting chromosomes, because that certainly wasn't random. And the reason it can't be random is when each of us were conceived, we had, say, a chromosome 1 from our mothers and a chromosome 1 from our fathers. And this had specific genes. And if you were to look into a cheek cell, a bone cell, a muscle cell, all of those cells need the chromosome 1 genes from your mother and the chromosome 1 genes from your father. So the division has to be organized so that one cell that has two copies of chromosome 1, that's the largest of them, chromosome 2 is a little smaller, chromosome 3, so that one cell that had two copies of chromosome 1, 2, and 3 now gives rise to two cells. Each of them have chromosomes 1, 2, and 3, etc. And so the cell that started off this process, which we'll call mitosis, had two copies of each chromosome. And I'll stress again in a little bit, that's a condition which we call diploid. And then this made two daughter cells. Each of the two daughter cells are diploid. So in mitosis, the mother cell divides and essentially produces two cells which are potentially the same. I mean, they could specialize afterwards. But mitosis is the process of copying. And this is especially important with the nuclear material. As such, then we split a cell's cycle into various stages. Now, embryonic cells are dividing very quickly. Most other cells take much longer, but it varies. And so, when you look at your DNA, how long does it take to replicate the DNA? Well, it can take 6 to 8 hours, but it could actually take much longer than that. So your origins of replication, you could use all of them or you could use only some of them, but that would then slow you down. So there's a lot of variations here. And some cells decide, I'm either going to temporarily or permanently leave the cell cycle and then go into this other phase, which we call G0. So while some cells, they're going to try to divide frequently. So my skin cells, constantly losing those. I constantly need new red blood cells or white blood cells. So whether it be skin cells in the epidermis or bone marrow cells, they have a cycle of cell division, and soon after they'll start a new cycle. Some cells will only divide, say, if I cut myself, all right? And then some cells never divide. Most of my nerve cells will never divide. My heart muscle cells will never divide. This is why I would really like it if I didn't have a stroke or a heart attack today, right? Because if I cut myself, that's bad, but I can make new skin cells. But if I have a heart attack and heart muscle cells die, I now have to live the rest of my life with less. If I have a stroke and nerve cells die, I now have to live the rest of my life with fewer neurons because these cells have entered a G0 phase. They are no longer capable of division. So some cells say, I cut myself, they can jump back into the cycle. Other cells have left it for good. Now, for those cells which are in this phase, most of the cell cycle is what we call interphase. So in the images that I had shown you on that PowerPoint, the chromosomes which were visible and actively dividing, that then occurs in this mitosis phase, which occurs at 1 hour. So if you've studied mitosis in high school, you might have looked at a fish embryo dividing or an onion root tip dividing. The mitosis, where you actually see the chromosomes, it doesn't take long. Most of the cell cycle is here. That's why you want to get actively dividing cells. There is a stage known as G1. That's the longest stage of the cell cycle. Here cells are growing because after they finish dividing, a cell is half the size of what the former mother cell was. So before it's ready to divide again, very often it's going to grow in size. So it's going to grow a lot, make a lot of new proteins, make a lot of organelles, because while the nucleus, there's a way to divide that evenly, what happens to the cytoplasm, it just kind of split it in half. But now this cell has half the number of mitochondria, half the number of, the amount of RER, et cetera. So it's going to make a lot of new things. Now, as I can discuss in another video on cancer, there are checkpoints along the way, right? You just don't go through G1 and then automatically go into the next phase. If you're not ready, if it's not appropriate for you to keep on dividing, then you stop. You don't keep on going until you have permission, and cells try to control cancerous growth that way. But if it is appropriate to divide, you can then enter the S phase, where you replicate your chromosomes. Let me get back to that. I have a video on why you would need to do that in just a minute. Once you replicate all of your DNA, so now you have 2 sets of everything that could be divided into 2 daughter cells. You then go through a G2 phase. G2 was, the G originally stood for gap, because people looking under simpler microscopes couldn't see anything happening, so it looked like nothing was happening. I think now it's more common to refer to it as a growth phase. This is when the final preparations for cell division is going to be made. So for example, in upcoming videos, you're going to see that there's this set of microtubules which enclose the chromosomes in what's called a spindle. Well, in this phase, a lot of the tubulin for the spindle is being made, so that when mitosis requires the spindle, that all of the components are there. The actual division of the nucleus then occurs in a relatively short period of time. And so what we refer to as prophase, metaphase, anaphase, telophase, that's occurring in this mitosis phase. Now, what usually happens is when the nucleus divides, so too does the cytoplasm, although that's technically a separate procedure called cytokinesis. And so now we're left with two daughter cells which have the exact same chromosomes after prophase, metaphase, anaphase, and telophase. Now, why do we need to replicate our DNA in S phase? Well, very simple. Like I said, my cheek cells, they need the genes on chromosome 1, 2, 3, and 4, so do my bone cells. If when I was an embryo years and years ago, if that first cell that divided into 2 did not replicate its DNA beforehand, then when it split into 2 cells, each of them would have had half the number of chromosomes. Then when it divided again to make 4 cells, then the chromosome count 10 would have gone down by half again, and then by half again, and then by half again. And so then soon, if I'm 100 trillion cells, then there wouldn't be any DNA left. And there certainly wouldn't be all of the genes from chromosome 1 and 2 and 7 and 9, et cetera. So the only way of ensuring that my cheek cells, my bone cells, et cetera, have all of the genes on chromosome 1, on chromosome 2, on chromosome 3, on chromosome 4, 5, et cetera, is if before a cell divides, there is an S phase where each of these then replicates. So now there's 2 copies of chromosome 1 you got from your mother, 2 copies of chromosome 1 you got from your father, and they will continue to adhere to each other. We'll see this as what we call a pair of sister chromatids. So one copy of chromosome 1 you got from your mother copies to be a pair of sister chromatids from chromosome 1 you got from your mother, which later in the cell cycle will then divide to be then separate chromosomes. Once again, the cytoplasm divides just kind of more randomly. But there is a very specific process to the division of the nucleus. Notice that these microtubule structures known as centrioles, there originally is one pair of these. They replicate and they move to opposite poles because if we're going to make 2 daughter nuclei out of this mass of DNA, where do the 2 start? We have to kind of have, say, like, hey, half you come this way, half you come this way. And the spindle will organize around these centrioles. Having an abnormal number of centrioles will promote cancerous growth because it can promote then abnormal numbers of chromosomes assorting to either side. Notice that this DNA will have to condense in prophase. I'll go through each of the phases more slowly. These now condensed chromosomes will then align along the middle of the cell. I've colored them different colors here because, like I said, we have chromosome 1s, 2s, and 3s, and we need all of our daughter cells to have 2 copies of chromosome 1, etc. So then, maybe 2 copies of the chromosome 1 are maroon, and 2 copies of, chromosome 2 are the light, blue, etc. Then there will be a phase called metaphase where they line up at the center of the cell along what's called a metaphase plate. I'll go through all of this more slowly in a second. Notice that the sister chromatids adhere to each other. Once again, they had copied themselves. So the mother cell had 1 copy of chromosome 7 you had from your mother. Now there are 2 copies, but they still adhere at what's called a centromere. In the anaphase, the centromeres separate, and now the sister chromatids can be pulled to opposite sides of the cell, and then we will then have them unwind to form 2 daughter nuclei. Notice that the cytoplasm is splitting at the same time. That's a separate process called cytokinesis. So there are stages in that last hour of cell division known as mitosis. And so now let me go through the first one, which takes the longest. All right, and so out of the phase of mitosis, if this is going to take an hour, notice that half of this is prophase, metaphase and anaphase only minutes, and so this is the longest phase. So if this cell has gone through G1, G1, S, and then G2, all right? Now what's going to happen is in this prophase, we find that the centrioles, they're going to have to replicate and move to opposite sides of the cell. The DNA is in a form that's not likely to be moved. All right, you can't move unwound DNA. And I talk to my students, so if someone said, I'd like you to move to a new apartment like in 10 minutes, you're not ready. Your stuff's all over the place. Your clothes are in these drawers, your books are over here, etc. If you're going to move, everything has to be packed in boxes. All right. So right now, this DNA is not packed in boxes ready to move. It's unwound. One nucleus in a human cell has 6 feet of DNA, 6 feet in a microscopic nucleus. That's not ready to move, especially since, when we went through an S phase, you actually duplicated to make 12 feet of DNA. So we have all this unwound DNA. It's not ready to move. So we're going to have to condense it into essentially tightly packed boxes. That's what chromosomes are. So when cells are not dividing, we don't see visible chromosomes. Instead, we see this unwound chromatin. Also notice we've enclosed this in a nuclear membrane, but we're going to have to package that up for later. So we put the nuclear membrane components in vesicles for later. So as prophase continues, we have chromosomes pointing every which way. But then starting from these centrioles, we have microtubules forming what's called a spindle that's going to attach to chromosomes, and they're going to move along this and start to order themselves, okay? So in early prophase, the DNA is this unwound chromatin stage, whereas by the time of late prophase, the chromosomes have formed as tightly wound DNA. Also, in early prophase, there was one pair of centrioles, but they replicated and then moved to opposite poles of the cell. The nuclear membrane in early prophase was intact, but notice by the end of prophase, we have dissolved the nuclear membrane and we will reform it later. We don't need a nuclear membrane because we're going to have these microtubules of the spindle, and notice that they are organizing around the centrioles. They are going to attach to chromosomes, and then chromosomes can then move along microtubules. Once again, there are proteins which can attach to a chromosome and can attach to the microtubules and move along it. So once again, here's in early prophase, there's one pair of centrioles. At late prophase, two centrioles at opposite poles of the cell. Some cells have decided they're not going to divide anymore, so they no longer have centrioles. They will be permanently in G0. So what you see here, this is the first part of cell nuclear division, known as mitosis. And this first phase is called prophase. This takes more than half the time of mitosis. We start off with a nuclear membrane, unwound DNA, one pair of centrioles, and no spindle. But by the time prophase is done, we have the spindle, two pairs of centrioles. The nuclear membrane has been packed away for later, and the chromosomes have formed from tightly wound DNA. So this is a very important phase in mitosis, obviously. I then just follow up with a couple of things here. So once again, the nuclear membrane encloses all of the DNA, but now we're going to have to move the chromosomes to make two piles, and the spindle's going to have to intrude in this area. So the nuclear membrane then has to be undone so that all of this can happen. And the next video will cover the condensation of the chromosomes. So we always have DNA, but we don't always have chromosomes. Chromosomes are the tightly packed DNA. Normally, when cells are not dividing, this DNA is in the form of chromatin. Now we can move this because there are regions of the chromosomes once it starts to pack together. So notice the DNA starts as this unwound chromatin, but we can now tightly wound it to form a heterochromatin as opposed to the unwound euchromatin. And so once we wind it very tightly, we now have what's called a chromosome. So the DNA has always been there, but at the beginning we called it chromatin, the unwound form that wasn't ready to be moved. Now we have a chromosome that is ready to be moved. Proteins can attach to it, can attach to the cytoskeleton and the nuclear skeleton, those lamin proteins. And so then chromosomes can be pulled to various points. So I go through some stages there. So this unwound chromatin is going to form tightly wound chromosomes. Proteins then bind to it, and then we have a cytoskeleton, but we also have a nuclear skeleton. And then proteins can move these chromosomes along the nuclear skeleton. And repeating a video from the cytoskeleton part, there are proteins which can link the cytoskeleton outside the nucleus to the nucleoskeleton inside the nucleus. And if these proteins then attach to chromosomes, then we can actually then move them and position them the way we like. And this is what's known as a link complex. So there's proteins both on the inside of the nucleus and the outside, which can connect. The mitotic spindle is made of microtubules, and this will organize around the centrioles. There will be fibers of an aster pointing away from the nuclear region, and then the fibers which then come in here, which, you know, in a sense form a kind of a cage to keep the chromosomes in one region, but also proteins attached to the chromosomes will then attach to the microtubules that they can then pull themselves along. And so you saw chromosomes moving to opposite sides of the cell. Well, these microtubules were the trackways that they were using. And so the tubulin protein, which makes up the microtubules, this was synthesized in G2, and then it forms the spindle in prophase. Okay. Once prophase is done, now essentially what we have to do is line the chromosomes up in the middle of the cell, because as we have pairs of sister chromatids, one of these has to go into daughter cell number 1 and the other one has to go into daughter cell number 2. So we just kind of have to say, all right, everyone hold hands, form a line in the middle of the room, and then let go of your partner's hands and move to opposite sides of the room, and then we're essentially done. So in metaphase, we go from having chromosomes in this unorganized format into a highly organized format where they line up along the middle of the cell, all right? And so now that everybody has a partner, all right, 2 sister chromatids attached by centromere, now that microtubules have attached and now they can move themselves and position themselves, now we can all line them up in the middle, all right? And so that all we'd have to then do at that point is say half of you go, you know, to the top of the screen, half you go to the bottom of the screen, and now we have 2 equal piles. Now, this is a little beyond the scope of this course, but this is really important because if cells don't do this stage well, they should stop. They should not then go to what's called anaphase. They shouldn't separate their chromosomes. And so this is a time where a cell can have a checkpoint to make sure if everything is okay. Because if it isn't, you could say, all right, well, don't divide. And if a cell doesn't pass this checkpoint, you can kill it, all right? Because the problem is, if too many chromosomes would go to the left or to the right, we would have potentially a cancerous cell, and we certainly don't want that. And so what you're looking for is tension. So you should have microtubules pulling one of the 2 sister chromatids on one side, and microtubules pulling sister chromatids on the other side in metaphase. And if we don't have that, this cell should stop. It shouldn't divide unless we can be sure we can pull equal numbers of chromosomes to the sides. So here, all right, notice that there's microtubules that are going to pull this sister chromatid. So this is one copy of, say, chromosome 1 you got from your mom, pull that down. Whereas these microtubules are going to take this copy of sister chromatid number 1 that you got from your dad, they're going to pull it up. If that's not the case, if both of these are pulling down, all right, then this cell can't divide, because then the daughter cell would have the wrong number of chromosomes and be potentially cancerous. So notice that the sister chromatids are tied together. There are proteins called cohesins, and we're not going to cut them until we're sure that the cell is ready to divide. Once we get approval by passing, say, a checkpoint, then we can cut these cohesins, and now we have anaphase, where the 2 sets of chromosomes are pulled to the opposite sides of the cell. Once again, very controlled. You don't want to make a mistake. If one of the 100 trillion cells of your body makes a mistake and starts on the road towards cancer, you could actually, this could cost your life. I mean, this is serious. So unless cells are lined up properly, or the chromosomes are lined up properly, you shouldn't go forward, all right, because mistakes are potentially lethal. If, however, there's appropriate tension, you can now pull the sister chromatids to the opposite sides of the cell. So once again, there are proteins which have wrapped these sister chromatids called cohesins. And if everything is lined up correctly, we're ready to cut that. And now the microtubules can pull the sister chromatids to the opposite ends of the cell. If we now have two piles of chromosomes, all right, one over here and one over there, well, we're done. I mean, that's what we wanted to do. And now we're ready for the final phase of mitosis. The final phase, telophase, is essentially the opposite of prophase. What did we do in prophase? Well, we took unwound chromatin and we condensed it to form chromosomes. So what's going to happen here? Well, you're going to take highly condensed chromosomes and then unwind it to form chromatin. In prophase, the nuclear membrane had to be broken apart and packaged for later. Whereas now we want to have a nuclear membrane surrounding this, so now it reforms. And so here you see the highly condensed heterochromatin, then unwinding to form, it was heterochromatin forming chromosomes, now it's unwinding to form the euchromatin once again. So after anaphase, we have two piles of chromosomes and the cells can then form nuclear membranes and then have two separate piles. Now, what usually happens, does not have to happen, but usually happens, is when the nucleus is splitting into two, then the cytoplasm can then split into two, and it doesn't really have to do so in an orderly fashion, it just kind of forms a furrow that splits the two. So that now we have two separate cells. Sometimes the nucleus splits, but the cytoplasm doesn't, and now you have two nuclei in one cell. So liver cells can do that, for example. Cells in fungi can do that. But in general, the last phase of mitosis, telophase, is accompanied by the division of the cytoplasm, which is known as cytokinesis. And if then we were to just, go back and see that here, here's a cell in interphase. And I know that's because in the nucleus, look, I don't see chromosomes. I just see unwound chromatin, all right? So these are cells in interphase. Once we've approved this cell for division, it then gets prepared to divide its nuclear material in an orderly fashion. So the nuclear membrane disappears. Notice I can't see like a circle of a nuclear membrane around these chromosomes anymore. The unwound chromatin forms chromosomes, and the centrioles replicate and then move to opposite sides of the cell, essentially saying, half of you chromosomes are going to get pulled this way, half of you chromosomes are going to get pulled this way. Once again, here you can see no nuclear membrane, the chromosomes are in disarray, and now some of you are going to get pulled to either pole. That's prophase. Once prophase is complete, now we need to line up the chromosomes in the middle of the cell along what's called the metaphase plate. This is only takes a couple of minutes. Metaphase, maybe takes 3 minutes out of the hour of nuclear division. Here you can see the separate microtubules of the mitotic spindle, and the centrioles would be here at the center of the spindle. Notice the chromosomes are lined up along the middle of the cell along that metaphase plate. If everything is going as normal, then what we do is we tell the centromeres to now become unattached from each other. We cut those cohesins, and now we can pull the chromosomes to opposite sides of the cell. So half of you are pulled this way, half of you are pulled that way. So in anaphase, these chromosomes then get pulled farther and farther apart from each other. Notice the microtubules in the center. And then finally, well, we're done. We have two separate piles, and now all we have to do is undo prophase. So these highly condensed chromosomes, then they can start to uncondense. Notice how they're starting to fade, okay? We can reform a nuclear membrane around this, and then the cytoplasm can start to split just by forming what's called a cleavage furrow and squeezing these apart. So notice there's still some connecting. So here we're just starting to form a cleavage furrow here, but the cytoplasm is still linked. Here we've mostly separated the cells, but there's still some adjoining material here. And here we're finally separating the cells. So the division of the cytoplasm is called cytokinesis. Okay. Now, once upon a time, you were one cell big. And that one cell divided by mitosis to produce two daughter cells, which divided by mitosis again to produce more. And so now you're 100 trillion cells of you. There has obviously been a lot of cell division to get from that one cell that was once you to what you see when you look in the mirror. And it has then been mitosis, which has given us epithelial cells and connective tissue cells and muscle cells and brain cells, etc. So mitosis, all right, these cells can specialize later. Mitosis is what gives us most of the cells of the human body. Once again, nerve cells, muscle cells, etc. So the daughter cells of mitosis can produce most body cells. There is one exception. The cells of which result from mitosis cannot be used for reproduction. So if these two individuals, all right, they decided that they wanted to start a family and they engaged in a deep French kiss and a cheek cell from one of them went into the mouth of the other and smushed together, all right, do they make a baby? Well, the answer is no, because this cheek cell has two of each chromosome. There's two of chromosome 1, two of chromosome 7, 2 of 13. I didn't include them all, it would be too many. But there's two of everything. And if that fuses with this cheek cell from this individual that has two of each chromosome, the resulting cell then has 4 copies of each chromosome. So cheek cells are great for being cheek cells, but they can't be used for reproduction, all right? And the same thing if we were to look at fibroblasts in connective tissue. They have 2 copies of each chromosome. You can't take fibroblasts from one person and fuse it to the fibroblast of another person. There would be too many chromosomes. Same thing for brain cells. So the cells which result from mitosis are great for to make muscle cells, bone cells, etc., but they can't be used for sexual reproduction. That requires special cells known as gametes, sperm which are made in the testes in males, and ova which are made in the ovaries in females. And notice that while there is a chromosome 1, unlike this cell, or this cell that has 2 of them, this one only has 1 of them. This one only has 1 of them, although there's I think that's 7. That's one of them. This one has 2 of those. This one has only 1. So a sperm has 1 of each copy of chromosome. An ovum has 1 of each copy of chromosome. And thus, when they fuse, then you can now get a new cell with 2 copies of each chromosome, a new embryonic cell called a zygote. All right. So sexual reproduction requires new special cells with half the normal number of chromosomes. We have 2 words for this. Cells which have 1 copy of each chromosome, we call haploid, all right? So all of the human genes can be found on 23 chromosomes if we're not counting the mitochondrial chromosome for right now. So chromosome 1 is the biggest, chromosome 2 is the second biggest, chromosome 3 is the third biggest, you get the idea. And so all human genes can be found on these 23 chromosomes. Some cells only have 1 set of this 23. They only have 1 copy of chromosome 1, 1 copy of chromosome 2, etc. And such a cell is called haploid. But most cells of the body are not haploid. Most cells of the body have 2 copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3, etc., and they are what are called diploid. So cheek cells are diploid, muscle cells are diploid, nerve cells are diploid, etc. Mitosis starts with a diploid cell and then produces 2 daughter cells which are diploid. So in mitosis, a mother cell which is diploid produces 2 daughter cells which are diploid. Mitosis is essentially trying to clone the mother cell. But that doesn't work for sexual reproduction. Sexual reproduction then is going to need a new type of cell division, not mitosis, which produces 2 diploid daughter cells from the diploid mother cell, but rather a new type of cell division called meiosis, in which the cells will divide twice. They will go through prophase 1, metaphase 1, anaphase 1, telophase 1, and then prophase 2, metaphase 2, anaphase 2, telophase 2. And now as a result, we have 4 cells after 2 rounds of cell division, and these cells then are haploid. So meiosis involves 2 rounds of cell division and results in haploid cells, which is different from mitosis. And if then meiosis produces a haploid sperm that has 1 copy of 23 chromosomes and a haploid ovum that has 1 copy of 23 chromosomes. Then if these 23 chromosomes fuse with those 23 chromosomes, then you have a new diploid cell, a zygote. So 2 haploid gametes then fuse to form a diploid zygote. So here, the ovum has 23 chromosomes. It is haploid. If it is fertilized, so here's a haploid ovum. If it is fertilized by a sperm, which then has its own 23 chromosomes, then we have 2 sets of 23. We now have a new diploid cell. So 2 haploid gametes then form a diploid zygote. Now, I'm going to start to wrap this up. So for students, say, in Anatomy and Physiology 1, we are nearing what you would need to know, in Anatomy and Physiology 1 in one of the introductory chapters. There's a lot more to learn, but obviously the chapter on reproductive systems and ovaries and testes can then get into it in greater detail. So for example, in the testes in a male, the cells will go through 2 rounds of cell division. Prophase 1, there's obviously some things going on here that you can pursue later or listen to the video. Now they're lined up in the middle of the cell. This is metaphase 1. They separate, that's anaphase 1. They then form 2 daughter cells, that's telophase 1. But then they start to divide again without going through an S phase. They go through prophase 2, metaphase 2, anaphase 2, telophase 2. So now they have 4 cells. But instead of having 2 of the big ones and 2 of the little ones, they now have 1 big chromosome, 1 little chromosome. They are haploid. So this is what's going on in testes. The cells then change their shape. They develop swimming tails, so that they can reach the ovum. But the testes in males are the only place in the male body where meiosis occurs, a type of cell division which produces haploid sex cells, the sperm. Now the same would apply in women to ovaries. This is where meiosis occurs to produce ova. It's a bit more complicated for a lot of reasons, and so I have videos in the reproductive system playlist. One of the things that you'll see here is instead of getting 4 gametes, you end up getting 1 gamete and then 2 or 3 cells which don't participate in reproduction, called polar bodies. So meiosis in females is a little more complex. And if you really wanted to, and this would be a bit more appropriate in, say, a genetics or a general bio 1 class, you could then compare prophase in mitosis, prophase in meiosis 1, prophase in meiosis 2, since there's 2 rounds of cell division. And so I have a number of videos on comparing the various stages of mitosis to the comparable stages in meiosis, if you're interested. But once again, for AMP1 students, this is probably a bit more in depth than you need to know, although the videos are here if you would like. And so for AMP1 students, meiosis is covered in much greater depth in Anatomy and Physiology 2 in the reproductive system chapter. And for AMP1 students, maybe the appropriate place to go from here is to say that while mitosis, is great, once upon a time I was microscopic, and I wouldn't be here, 100 trillion cells later, if it wasn't for the mitosis, which turned 1 cell into many. Mitosis can be a problem because here are cells of a cancer, of breast cancer. Here are cells of leukemia. Here are cells of an ovarian cancer. So cell division has to be controlled. Uncontrolled cell division, that's potentially cancerous growth. And if 1 of 4 Americans gets cancer, I had a very, very minor skin cancer thing, which is removed very quickly, not a problem, but nevertheless, that's, frightening. Any of us can have cells that start to divide without control. So then mitosis gone awry is a serious health problem. And then I then have another video that AMP1 students might want to follow up with on what causes cancer.